If anyone can't join us, then they can speak. I should be seeing 17 people on here. Taking the dub. Oh. Whenever I open the chat, it is freaking out on me. I don't know why. Okay. I am missing 2 people. Who are we missing? The chat is freaking out on me, so if anyone is trying to message me, this is gonna get complicated.
Somebody said that they're joining, if you can't see it.
Wait,
They
say that
just
again?
joined. It was Trace.
I'm in.
Yeah. Mhm.
Okay. Did we lose people? Just saw 15 went down.
Oh,
We lost a few.
Okay, let me turn off the music for a second, because I am turning into a grandma. What is happening?
we're missing 2 people.
Yes, we are
Oh.
missing 2 people. Okay, so we have Trace, Kiara, okay, Nicholas. Who are we missing? If anything, I think you can still join in. But let's get started so we can get through as many questions as we possibly can. Okay, so you guys have an exam coming next week, so this is why I'm doing this. What term is used to express the idea that several genes exert influence over the same characteristic? Yay! I don't know why everyone keeps choosing co-extension. My past 2 classes had some students do that too. I don't even know where I got that term from, but it is gene interaction, so good. Guys, we never talked about this
Whoop,
word.
whoop.
Wait, what happened?
I said whoop whoop, but it's irrelevant.
Okay. So good. That's the definition for the gene interaction. So very good, very good. I like it so far. Good. Okay. In codominance, both phenotypes are expressed in heterozygotes. Yay. Good. Codominance. It's when both phenotypes are expressed. An example for that would be, what's an example of codominance?
Unknown.
Wait, say that again?
I have no clue.
AB blood type, right? Because
Yeah.
it's showing both A and B antigens. Okay. So we good with codominance? Codominance, any of the types of dominance, you look at one gene pair, right? And it's looking at both the alleles. Okay. Good job, guys. I'm proud. In incomplete dominance, the heterozygote shows an intermediate phenotype between the two homozygotes.
Look how the music just sounds really distorted to us.
Yeah, that's my favorite part too.
Good. Incomplete dominance, it's always a mix or an intermediate of the two phenotypes given out, right? So good, good, good. Love it. Good. Okay. With incomplete dominance, a likely ratio resulting from monohybrid cross would be so yes, it is 1 to 2 to 1. Because incomplete dominance, you actually see a distinct heterozygote phenotype. So guys, this is for dihybrid crosses. This is actually epistasis one, depending on it's because it's a modification of this guy. When you're thinking monohybrid cross, remember it's one gene, right? You're looking only at one gene. These two are involving two genes, this ratios. So now we're down to this guy, right? We have 3-to-1. That's a regular monohybrid cross. But when incomplete dominance is at play, you get a 1-to-2-to-1 ratio, right? Because the ones are the homozygotes, and then you have 2 heterozygotes, right? That have a distinct phenotype. Does that make sense? Great. Okay.
Mhm.
When a cross is made, complete linkage is shown when offspring show parental and recombinant genotypes. Oh, okay. No, something in the chat. What's going on? Okay, the chat hates me today. I don't know why. But what's going on, guys? Okay.
Not much.
Nope. What were you about to say, Mia?
Not much is happening in the chat. You're fine.
Okay. So complete linkage, they're so close together that you cannot see any recombinants. So that's why the answer is false, because you do not see the recombinant genotypes, right? Does that make sense?
So when it's complete linkage, you won't see the parental phenotypes, right?
You will only see the parental groups.
Okay, so you won't see the recombinant groups.
They won't see recombinants because they're so close together, like maybe this close. There's not much room in between to actually have some recombination going on. Does that make sense? Yes? Okay. Wonderful, wonderful, wonderful. The phenomenon in which a single gene pair influences the development of many phenotypes is called, I don't know if we actually went over this in discussion itself at least. Okay, yes. Pleiotropy is the answer to this guy. So when a single gene influences many phenotypes or gives rise to different phenotypes, that's called pleiotropy. The confusion here with epistasis is a gene controlling another gene. Just one gene that gives rise to a certain phenotype, not many different ones, right? Does that make sense?
Yeah.
Great. Okay. When a cross is made, independent assortment is shown when offspring only show only parental genotypes. True or false? Okay. So it's the opposite of the other one, right? So we have, so when you have independent assortment, there is room for recombination, right? So you would actually see recombinants and parental groups. We clear on that kind of idea, I guess? When they're linked, you see the parents only. And then when it's like, if it's not considered linked, or if they can actually independently assort, they will have parental types and the recombinant types, okay? Two genes that are separated by 20 map units show a recombination percentage of 20%. Wow, you guys are quick. Okay, you guys are pretty much answered that. Wow. Good job, guys. To everyone who put true. So guys, remember, one map unit equals 1% recombination. When you have 20 map units, it has 20%, right? Be good. Well, considering majority of you put that, I'm assuming yes. Okay. A condition in which one gene pair masks the expression of a non-allelic gene pair is called blank. Good. Guys, whoever put codominance, remember the 3 types of dominance is focused on alleles, not gene pairs. Epistasis is the one that controls gene pair, one gene pair controlling another. So these guys are just one gene pair comparing 2 alleles. Remember that part? Just want to make sure. Okay. The mapping of an individual chromosomes is called, what is this thing? Good. So that's the technique, at least, to figure out the mapping of the chromosomes themselves. It's karyotyping. Okay. Traits controlled by genes located on the X chromosome are blank traits. Good. Okay. So X chromosome, sex linked, that's the direct connection right there. Anything that is not a sex chromosome is an autosomal chromosome. Polygenic just means multiple traits, multiple genes. So that has nothing to do with the X chromosome per se. So good work. Okay. What is the useful purpose of chromosome maps? I don't know if we covered this or not, but give it your best shot. Good. It is to map genetic diseases. So the others, so chromosome maps or karyograms or karyotyping, that is essentially how we can map genetic diseases. It'll be covered more in the next topic that we cover with chromosomal mutations, that you can actually see the genetic diseases or genetic alterations using these chromosome maps, okay? Don't worry about this question too much. I think it was supposed to be part of the next session, but yeah. Okay. The prodinor mode of sex determination. Oh my. Okay. I am glad it's tied out at least in one class, because everyone else put the XXXY scheme. So the answer is XXXO. Lena will be proud in that at least it tied out in one class. So good. So what mode is this guy? XXXY. Do you guys remember? No? Okay. So what is this? This guy, right? The ligase mode. That is yay. Okay. So guys, remember, prodinor mode is this XXXO scheme. Ligase mode is XXXY. Okay. Let's not upset Lena. Lena the nematode gets very picky about those things. Okay? The chromosomes that are not involved in sex determination are good. Autosomes. Okay, analogous and homologous are just telling you the relationship between different chromosomes. It has nothing to do with sex. So those are just saying the relationships. Autosomes are the ones that are not involved in this. So just keep that in mind, okay? Sex determination varies between animals, true or false? Cool. Yep. So again, that's why we have the different modes, right? So sex determination does vary between animals because they have different chromosomes that are involved in that determination. Good. In humans, sex determination generally depends on what? Good. All of the following genes are involved in male development except,
This is where I'm lost.
wait, say that again?
I've officially lost as of now.
Oh. Good. It is the XIST gene. So these guys are all some way, some shape of male development. XIST is one of the genes that is involved in exon activation. I don't know if I covered that with you guys, but that is one of the major genes that are involved there. Does that make sense? Okay. Heterogametic sex determines the sex of the progeny. Heterogametic as in like heterozygous with the sex chromosomes. Yep, it is true. Um, an organism containing 50 chromosomes will produce what? So ignore pairs here. That was a typo. Good. So again, 50, instead of the pairs, look at 50 chromosomes. You would end up with gametes that have 25 chromosomes, right? Because it's split in half. Okay? "48 chromosomes are present in a somatic cell of an individual with both Klinefelter and Down syndrome." So in this case, Down syndrome is 3 copies of chromosome 21, okay? So true, false, what's going on? Oh, okay, why are we thinking false?
I don't know.
Okay, so with Klinefelter, what's the genotype for that? What's the difference from a regular person? Two X's and a Y chromosome, right? So it's an XXY for Klinefelter. So that already adds on one more, right? From the regular human. Regular humans have 46 chromosomes, right? On top of that, this person has Down syndrome, which has 3 copies of 21, chromosome 21. So you're adding an extra copy of Down syndrome. So with that, you have 48 chromosomes, right? So that's why the answer is true. Does that make sense?
Yeah.
Cool. Yay. Okay. How many Barr bodies does a cell from an individual with the karyotype 47 or XXX contain? It's also called triple OX. Yep, it's 2. Where did we get 3 from? So guys, remember, Barr bodies are inactive X chromosomes in a set. One remains, so in a case like this, or any case with multiple X's, one remains active, the remaining X chromosomes end up inactive, right? So in these cases, you have two bar bodies because you have two inactive X chromosomes. Are we clear with bar bodies? Yes?
Yeah.
Okay, good. Good. Okay, 3 more questions. Calico cats are usually female, but occasionally a rare male calico cat is observed with the karyotype of XXY. True or false? Yay! So yes, that is true. Calico cats, remember, they occur because of X inactivation, the randomness of it, and the mosaic pattern that comes out of it. So for a male calico cat to occur, you need another X chromosome to show that specific alleles for that specific trait, right? So that's how you end up with XXY for a calico cat with males.
Okay, Mommy, I have a fire alarm going off, so I have to go. I'm really sorry. I'm gonna go.
Oh,
Oh wait, I'll try to do that.
all
Bye.
right, teaching.
That's tough.
Now I can get third place uncontested.
You know what? In that case, I am staying.
Feel bad. Oh no. No, be safe.
Be safe.
Take the computer with you.
That too. Okay.
I'm staying. You're gonna come with me.
But are we cool with the calico cat? Cool. Okay. Paternal X is always inactivated for dosage compensation in placental mammals. True or false? Wow. You guys split even. Okay, so what do we know about X inactivation? We know that it's random, right? So it doesn't mean that the paternal X is the one that is always getting inactivated. It could be the maternal, it could be the paternal, who knows, right? So that is why the answer is false, because this is kind of following Lyon hypothesis, where they say that it occurs at an early state. It also is random. So that's what's happening with that. Does that make sense? Good, okay. Which of the following human genetic conditions is missing a chromosome? Good, it is Turner syndrome. Jacob syndrome has an extra Y chromosome, right? X, Y, Y, okay? Klinefelter, it's X, X, Y. And then triple OX, that's just 3 X's in a row, okay? One last question. SRY is the major control center of X inactivation in mammals. Oh. Guys, where is the SRY, or what is the SRY? Do you guys remember what the SRY was?
No, I just guessed.
I like the sex determining region thing.
Yes. Where
Yeah.
is it though?
I don't remember.
SRY? Y chromosome, right?
Oh,
So, okay, so remember with, I don't know, did I cover this with you guys? I have no idea anymore. I have 3 sections and everything's becoming a blur at this point. But, okay, anyways, the SRY is the sex determining region of the Y chromosome, okay? So that is specifically on the Y chromosome. The major control center for the X inactivation in mammals, that's called X inactivation center, or XIC, okay? I think it's on the PowerPoint as well, so make sure you guys know that too, okay? So that is pretty much all I got. Woo! Yay!
I didn't take the dub.
Okay, this can stop.
I'll
right
take second place. That was great.
So, I hope this helped. Do you guys have any questions? Concerns? Do you guys like this format of discussion?
I like this. Yeah, I think
It's
it works
good.
well.
Because I might do it more often if you guys actually like it.
Yeah, it's fun.
It's top tier.
And it does get you guys to participate more, because I've noticed with the past couple of discussions, I hear the same 2-3 people talking. So, but if you guys don't have any questions, that's pretty much all I got for you guys. Let me just double check something before I let you guys go. Okay.
Bye.
Bye, guys.
Thank you.
Bye. Thank you so
Bye.
much.
Thank you.
Not
Take
a
care.
problem.
Bye.